From the JAMA Network, this is JAMA Oncology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Oncology.
Hello, and welcome to JAMA Oncology Podcast. I am Dr. Vivek Subbiah, medical oncologist and chief of early phase drug development at the Sarah Cannon Research Institute, Nashville, Tennessee, and also the JAMA Oncology web editor. Today, we have Dr. Kantarjian talking to us about the Olverembatinib study that was published in JAMA Oncology. The Olverembatinib study is a phase 1b/2 randomized clinical trial in CML after failure of tyrosine kinase inhibitors, including Ponatinib or Asciminib. Dr. Kantarjian is a professor and chair of the Department of Leukemia at the University of Texas MD Anderson Cancer Center, where he's also the Samsung Distinguished Leukemia Chair in Cancer Medicine. He needs no introduction in the heme malignancy and leukemia space. Dr. Kantarjian has received innumerable prestigious honors and awards. Dr. Kantarjian's research and collaborations were the basis for the FDA approvals of over 20 drugs in leukemia. He is an author on over 2,000 peer-reviewed publications. Dr. Hagop Kantarjian, it's a pleasure and honor to have you here.
Likewise, Dr. Subbiah.
So let's dive in right to this paper. What inspired you to investigate the pharmacokinetic profile and efficacy of Olverembatinib in non-Chinese residents, specifically for patients with relapsed or refractory chronic myeloid leukemia and Philadelphia chromosome positive acute lymphoblastic leukemia? Can you tell in your own words for our broad audience the study's motivation?
Because we have an audience which may not be well versed in leukemia, I'm going to take a moment for a background. So in chronic myeloid leukemia, the year 2000 was a turning point because this was the year where we discovered the activity of imatinib, and we started treating patients with imatinib, which was the first generation tyrosine kinase inhibitor. And later on, we developed additional TKIs. So there was a wave of a second generation tyrosine kinase inhibitors, including dasatinib, bosutinib, and nilotinib. And then the third wave, focused on what's called third generation TKIs, which were active among patients who develop resistance because of a T315I mutation. And there were two kinds of third generation tyrosine kinase inhibitors. The one exhibited or highlighted with Ponatinib, and this was a drug that is the most active of the BCR-able TKIs, but it is also toxic. And this is a drug that targets the same able kinase domain and inhibits it. Then there was the development of what's called STAMP inhibitors. And STAMP inhibitors selectively target the able myristoyl pocket. And the example of this is a drug called Asciminib, which has been FDA approved for later line therapies of chronic myeloid leukemia. So as of today, we have the first, second, and third generation TKIs. Among the third generation TKIs, we have two potent TKIs which act at different sites, either the able kinase domain or the STAMP site. Now, the issue is Ponatinib is extremely potent but toxic at the FDA-approved dose. Asciminib is potent. We do not know whether it's as potent as Ponatinib or not, but we know that it is active and so far safe. But what we want to do is develop additional TKIs in both kinds of subsets. So we end up with the TKI that's the most potent, but also the safest. And this is the genesis for developing Olverembatinib, which is a drug that was tested in China and has received FDA approval in China to treat patients with CML with T315I mutation, as well as patients who have failed 2 TKIs or more. Because the Chinese population may have different pharmacokinetic profiles, we wanted to investigate the new drug in a more heterogeneous population, which led to this multi-institutional study. And in this study, we accrued so far 80 patients, including 62 in chronic phase, and the others were in transformation. And what we found out is that in patients with chronic phase, the drug was highly active. So the incidence of complete cytogenetic response was 61%. The incidence of major molecular response, which is a very important molecular endpoint, reducing the disease at a 3-log level, the major molecular response rate was 42%. And we found that the incidence of complete cytogenetic response and major molecular response was the same in patients with or without a T315I mutation. But then we come to a very important question, which is the following. Before Olverembatinib, when we took patients who were resistant, truly resistant to Ponatinib, and we gave them Asciminib, the incidence of response was 10%. If we took patients who were resistant to Asciminib and we gave them Ponatinib, the response rate was 30%. So we asked what would happen if we take patients who are either failures on Ponatinib or Asciminib, and failure can be true resistance or toxicity, and we give them the Asciminib. So among 26 patients who were exposed to Ponatinib, 15 had a complete cytogenetic response. The complete cytogenetic response rate was not low. It was not 10%. It was 58%. And when we took patients who were resistant to Ponatinib, there were 19 such patients and 10 responded. So the response rate was 53%. Now we said, well, this is very good with Ponatinib. Let's see what happens with Asciminib. And because Asciminib is a newer TKIs, we had less patients. So we treated 10 patients who were Asciminib exposed, and 6 of the 10 had the complete cytogenetic response. So a cytogenetic complete response rate of 60%. And among the 8 patients who were truly resistant to Asciminib, 4 achieved the complete cytogenetic response. And the drug is safe. So what we find is this new third generation TKI, which targets the same way as Ponatinib. So it targets the Abl kinase domain. That drug is highly effective even in patients who failed multiple TKIs, with a complete cytogenetic response rate of 61%, with a major molecular response rate of 42%. We find that the response rate is equivalent in T315I mutated or in the non-T315I mutated. But I think the most important finding is the drug is still highly active in patients who had previous exposures to either Ponatinib or Asciminib, and among patients who were truly resistant to either Ponatinib or to Asciminib.
Well, thank you so much for the broad overview. So given the favorable pharmacokinetic profile and the strong anti-leukemic effect that you just observed, how do you see Olverembatinib fitting into the current landscape for CML and Philadelphia positive ALL, especially for patients who have failed multiple TKIs?
So in chronic myeloid leukemia, what we would like to do is have an FDA approval. So we already launched a study in third-line therapy where we're going to compare Ponatinib to a standard of care, which is Bosutinib. And because we don't want to spend many years in development, what we have chosen is a surrogate endpoint of long-term outcome, which is the 6 months major molecular response. We're hoping that the study which has opened will complete the accrual within 6 to 12 months, because there's a lot of enthusiasm in exploring this new highly active and safe drug. And we're hoping that we'll get an approval in third-line therapy. Beyond this, what we would like to do is explore it not only in third-line therapy, but in second-line as well as in front-line therapy. Now, in the frontline therapy, the study is going to be very important because, as you know, all the TKIs produce the same survival rate. The patient's survival has become so favorable that the 10-year survival rate is about 92% if you look at a relative survival rate. So you cannot improve on a 10-year survival rate of 92% unless you put tens of thousands of patients and follow them for 10 years. What we're going to do is look at the incidence of durable deep molecular response, which is the surrogate endpoints for treatment-free remission. In addition to improving the survival, what we want to do is potentially stop the treatment in these patients, what we call a status of treatment-free remission, which is equivalent in my book to a true cure in chronic myeloid leukemia. What we would like to do is do a frontline study where we show that the incidence of durable deep molecular response is significantly higher with Olverembatinib than with all the other TKIs. And then we have hope to have a highly effective, very safe drug that will induce a high rate of treatment-free remission. Today, if you look at patients who achieve a treatment-free remission, that incidence is between 20 and 40%. We would like it to go up to 60 or 80% so we can have a treatment in frontline chronic myeloid leukemia, which has a potential of treatment discontinuation without the CML coming back. And I think Olverembatinib may fulfill this dream in the future. Now, as far as Philadelphia positive ALL, as you know, now we're piloting studies without chemotherapy, and this is with a combination of Ponatinib and Blinatumomab, which is a CD19 bispecific T-cell engager, and we're getting fantastic results. But the question is, can we have a drug safer than Ponatinib? So we're going to start exploring the combination of Olverembatinib and Blinatumomab in Philadelphia positive ALL, initially in the salvage and then in the frontline. What we are told from the Chinese experience is very reassuring. What they are telling us is in China, there were hundreds, if not thousands, of patients with Philadelphia positive ALL who have been treated with Olverembatinib, but I have not seen any published literature on this. So we need to get our own experience in the United States and in multi-institutional studies to try to establish Olverembatinib as one of the major components of non-chemotherapy curative approach to Philadelphia positive acute lymphocytic leukemia.
You mentioned about BiTE. I think for our broad audience who are listeners, they may not know what is BiTE. A BiTE is essentially a bispecific T-cell engager. It's a laboratory-made antibody that helps the body's immune system attack cancer cells. So how it works is that the so-called BiTEs bind to two different proteins on the surface of cells, bringing T cells and cancer cells close together so that the T cells can kill the cancer cells. So BiTEs are being investigated in many types of cancer. The success has been in acute lymphoblastic leukemia. We've learned a lot from that. Now we are using that in multiple myeloma lymphoma and solid tumors like small cell lung cancer. Again, more number of studies are going to be published in this space. So going back to the current topic of the day, Dr. Kantarjian, so can you elaborate on the potential molecular mechanisms by which Olverembatinib overcomes resistance to other TKIs like Ponatinib and Asciminib? And how does it compare to the mechanisms of other third-generation TKIs?
So we do not understand the full mechanisms of resistance, but what we know is that patients who become resistant to imatinib or to the second-generation TKIs, they can develop mutations at the Abl kinase domain site. Now, why would Olverembatinib work in people who are becoming resistant to first, second-generation TKIs and Ponatinib? I think it's an issue of increased potency, plus the possibility that Olverembatinib may have a multi-kinase effect that we have not fully explored. So, for example, in the experience from China, and I have zero experience yet in the United States, what they are telling us is that Olverembatinib may work in patients who are not Philadelphia positive, in patients who are Philadelphia negative ALL or pre-BALL. And I do not have a full grasp of why it would work. But if it does, this will open a whole new venue of research in acute lymphocytic leukemia outside the Philadelphia positive ALL. Now, in the patients who develop resistance to Asciminib, we are seeing mutations in the STAMP domain, but we are seeing also mutations outside the myristoyl domain. So this is still a puzzle in evolution, a question in progress where we're trying to understand the mechanism of resistance. So there are two kinds of mechanisms that we know. The first one is related to mutations at the attachment site of these drugs, be it the Abl kinase domain or the myristoyl site. And the other one is simple resistance that we do not quantify yet, and it could be overcome simply by having a much more potent BCR-Abl kinase inhibitor, such as Olverembatinib.
Could you discuss the most common adverse events observed and how they were managed during the trial?
So far, we have not seen much of a serious side effects. The two things that we see are pigmentation on this. These look like dark moles, but they are very tiny and they are not bothersome to the patients. And we've seen occasional reports of hypertension, sometimes nausea, sometimes aches, but we have not seen any of the serious issues that we've seen with other TKIs. So far, the incidence of pancreatitis, inflammation of other organs like pericarditis, myocarditis, pneumocystis, hepatitis, nephritis, we haven't seen any of those. There's not much of the GI toxicity or the nephrotoxicity. And these are the issues that we've seen in the past with the TKIs. So I'm hopeful that with a much larger experience, outside of China, we can confirm the same safety reports that were reported from the Chinese experience. And my understanding is in China, there are thousands of patients who have been treated. So we're hoping that we will get soon to hundreds or perhaps over 1,000 patients treated outside of China, mainly in the United States and Europe, to be able to start reporting on the side effects of the drug and report, hopefully, that the drug is quite safe, as was experienced in China and as was experienced in the first 80 patients that we're reporting in JAMA Oncology.
So what are the next steps in the clinical development of this drug, Olverembatinib, and any closing statements?
So as I mentioned, we'd like to develop Olverembatinib as later line therapy, both third line and second line therapy. We would like to develop it as frontline therapy of chronic myeloid leukemia to achieve a high rate of treatment-free remission. In Philadelphia positive ALL, we're starting to pilot studies with Olverembatinib and blinatumomab in Philadelphia positive ALL. But I am very intrigued by the possibility that the drug could work outside the context of Philadelphia positive ALL, namely in the Philadelphia negative pre-BALL and perhaps T-cell acute lymphocytic leukemia. So we're starting pilot trials along these lines to be able to understand whether the drug will have a role outside the Philadelphia positive entities.
Thank you so much, Dr. Kantarjian. It was such a pleasure talking to you.
Thank you very much for the opportunity, Dr. Subbiah.
I'm Vivek Subbiah, and I've been speaking with Dr. Hagop Kantarjian from the University of Texas MD Anderson Cancer Center about Olverembatinib after failure of TKIs, including Ponatinib and Asciminib. This episode was produced by Shelley Stephens at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. Thanks for listening.